A very good afternoon to everyone. Namaste, namaskaram. I would like to thank Professor Jagannatha Rao and the President, all the organizers of this meeting, Indo-French or SAFIPRA meeting that is there. And as Dr. Alex mentioned, it is not just a conference, it's a workshop or an intellectual meeting, basically. So that's that. I am trying to present some of our experiences in understanding the neurobiology of Parkinson's disease. And I do not have any financial disclosures. Yes, of course, I am the president of International Society of Magnetic Resonance in Medicine India chapter, and also vice president of National Magnetic Resonance Society, two of the main societies which represent MRI. So I may talk a little globally rather than only about my institute. My perception of this talk may be, or our understanding is trying to go from molecules to humans. And as I said, there are a number of MRI units per million population is just 0.21 in India, much, much lower as compared to many of the countries above. And the left side is the statistics from the government. Basically, if you know that MRI also comes under PCPNDT Act over the last couple of years. Earlier it was not, but last 5 or 6 years we have to register MRI also. And so the number of scanners as per the official documents is 5,000. But then I think there are 2 states which has not made this mandatory, so the MRIs are not registered yet. So according to the information from various vendors, there are 8,000 1.5 Tesla MRIs in the country, around 600 to 700 3 Tesla MR scanners, and then 0.3 Tesla to 1 Tesla, there are around 50 scanners. And the distribution of MRIs across the country, especially for the research, I have tried to depict. It is localized, or it is just packets which are there. And of course, today we are represented with various people from various institutes over here. Coming to the main topic, this is one of the old slides that I try to still use, that Parkinson's disease has both motor and non-motor symptoms, and many of the non-motor symptoms are very, very less explored. And the prevalence of PD was around 4.1 in 2005. And 2016, it's around 6.1 million. And the geographical distribution is also alarming. One reason may be the aging population, but others, it is because of the usage of various factors, environmental factors that are associated with that. And along with Parkinson's, there are, the very first time when I went to a neurologist working on that and was saying, I want to collaborate with you. Professor Madhuri Behari said, can you distinguish Parkinson's from multiple system atrophy, which has similar tremor, and then progressive supranuclear palsy, which also has some of the overlapping symptoms. So she said, can you differentiate this? That was the first task given to me. She had put a DM student on that, and subsequently a PhD. And then our research started on multiple aspects. So we started exploring using metabolic information in vitro and in vivo, and then functional information using motor task, cognitive task, resting state, and then structural MRI looking at the volumetrics, and then the diffusion tensor imaging based data. And for the talk that I am going to present today, we collected serum, saliva, and urine from the subjects, and this is how the sample is prepared for NMR. And we did a simple 1D experiment, and of course we did 2D experiment whenever we are not able to isolate any particular peak or assign a particular peak. And we have followed various publicly available as well as paid software for the analysis of the data. And we also estimated the concentration of the metabolites and did a detailed analysis. I'll be presenting around the work of 7 PhD theses and 1 DM thesis in this work. So as you see, we got this NMR spectrum from urine samples, and you can see the black one is in the healthy and the red one is in the PD subject. So it is, as I said, hydrogen, and so 0 to 10 ppm, the graph is there. And then we were able to distinguish PD from healthy based upon the urine metabolites. And of course, what are the metabolites that are very significant, starting from ornithine, acetate, leucine to tryptophan. So these are very significant metabolites that were in PD as compared to healthy controls. And of course, you see the heat map which says which is high. The red one is high and the green one is low. So the trend changes for a couple of metabolites over here, like valine, all those things. And then among them, the most significant ones with your p-value over here. So you see that ornithine, phenylalanine, isoleucine, beta-hydroxybutyrate. This is one of the metabolites that we could narrow down, because later we found that the gut microbiome can be actually investigated if you try to correlate this. And then tyrosine and then succinate. And of course, we also tried post hoc whether we can differentiate early PD, advanced PD, using any of the metabolites that we could. And these two provided that it could still be, like ornithine and beta-hydroxy, we could distinguish early PD and advanced PD from the healthy controls. Of course, among themselves, we could not. So the results from urine, how it is affecting various pathways, basically, and we see that glutamine and D-glutamate metabolism is downregulated, and the tryptophan, basically this biosynthesis is upregulated, and there are various other which are modified in the patient. And this same plot in a different way, we had to do a little rigorous study. This second one which we started, and collecting saliva sample I thought was the simplest. And when I asked the student, she said it is the most toughest. So we managed to collect, especially from a Parkinson's patient, where the salivating itself was a task in them. And we had collected saliva samples from 76 patients, and it was in 50 controls that we did. And interestingly, the student, after a couple of years abroad and elsewhere, has joined back as a woman scientist, and she is continuing the work. So, because when we asked her to do this work, she was very hesitant, and now this is most highly cited paper that was there. So this was, already it has crossed 60 or 65 citations. So here, except a small overlap, this was much better, and if you go to a 3D representation, we could as well see many of the metabolites very distinct from healthy. That is something marvelous, and so we found that the metabolites, the histidine, tyrosine, phenylalanine, acetate, propionate, isoleucine, valine, these were very much different, and the p-values were very significant. And along with that, we also found TMAO to be affected. It was actually a little higher than that in healthy, and we thought that TMAO concentration is higher because of the assimilation or the secretion, and later we found out during the analysis that it is because of the malabsorption that TMAO is higher in the saliva samples. So these are the summary of the whole thing, and the same in representation. And of course here, we could have some more metabolites which were able to differentiate early PD and advanced PD with respect to healthy. And as I had mentioned, so between early and advanced, we could not still differentiate. That is one area that we are still working on. And we collected serum from the subjects, And again, TMEO was one of the most important marker over here. And then the glutamine, glycine were also significantly altered. And when we did the AUC cutoff analysis, again, these metabolites were very significant. And TMEO was the only metabolite that could differentiate early and advanced PD with respect to the control in the serum. But of course, as I said, between the EPD and advanced PD, we could not differentiate. So what we also parallelly had done is we created a Parkinson's disease model using 6-OHTA. This was a unilateral model where the injection was given in the striatal area, and like basically 2 injections were given, and we also had a saline injected sham group and non-injected control group. So 3 groups were there, and we followed up the work at baseline, 3 weeks and 7 weeks post-surgery. And the serum was collected from the optic vein, and we also had carried out MRI in this, and we also carried out relaxometry studies at these 3 time points over here. And we have a 7 Tesla small animal imaging facility, so we did this using that. And yes, there is a partial overlap in the 2D spectrum, but in a 3D, yes, we could differentiate the metabolites very well. So what we tried to do was basically, as you see, the between 3rd and 7th week was our main interest, but basically how the disease progresses, that's what we wanted to know. And but then interestingly, we could not get any significant signature between that. But then yes, baseline with versus 3rd week and baseline versus 7th week, we got significance. And so the glutamine, alanine, choline. So these 2 metabolites were significant between 3rd and 7th week. So only lactate and choline were different between the 2 time points. Another student worked on the Parkinson's with normal cognition, Parkinson's with mild cognitive impairment and healthy controls. And of course, because of the consistency, we tried to focus on only 4 metabolites: N-acetyl aspartate, total N-acetyl aspartate, total creatine, total choline. And this was acquired using 2D chemical shift imaging in the brain. And the left hemispheric frontal lobe NAA and creatine were very much different between healthy and PD. However, this just had a trend and not actually significant in the right. So that means even the metabolic, the frontal neurodegeneration pattern is a little different in the left and right hemispheres. Now, summarizing this part of the work. So when we tried to combine human biofluids, urine, saliva, and serum, and then the serum from the rat, this is the combined metabolic information, and we found that the metabolites derived from gut microbiome basically were elevated in PD as compared to the healthy controls. So the metabolic pathway that are affected basically, so we are trying to see like whether it is TCA cycle or urea cycle. So we are able to do a detailed study, and yes, the next step would actually be gut microbiome that we are trying to see. So here the conclusion that I am trying to do is basically the malnutrition in patients with PD. That is something that we are trying to see how we can understand that particular part. Now, just switching gear, the second one is the functional MRI and the MRI that I would be talking about. And in functional MRI, I thought I'll just introduce the concept of BOLT, the blood oxygen level dependent that happens. And typically when there is a task, you have the activated, basically the deoxyhemoglobin decreases and oxyhemoglobin increases. I would differentiate in this way. So when there are some processes which require some neurons firing, so you have increased oxyhemoglobin and deoxyhemoglobin. Basically, the susceptibility difference is very less. And we are trying to map this very small value. Of course, as you go on higher fields, the signal strength varies, and it is also dependent on the task. A motor task would give more than 10% variation. Motor task or occipital, any visual task will give you more than 10% at 3 Tesla, but it will be reduced to a little less than half at 1.5. The cognitive processes, it varies from 3 to 11%, depending upon the intensity of the cognitive process that are going to fire the neurons. Now, functional MRI, I have heard many students and everybody talking. It's an indirect process of depicting the blood, the activation in the brain. It is not a direct process. So that is one caution that you have to always have. It is a combination. The change in signal intensity is going to be difference in the cerebral blood flow, cerebral blood oxygenation, and cerebral blood volume. So it is all the three that is going to play a role, and not just cerebral blood oxygenation. And this signal basically gets transformed to T2, or I would say T2 star, because gradient echo images depict a better representation of the bold signal. So we use a sequence called echoplanar imaging to map that. And the first fMRI report was in 1990 by C.G. Ogawa. So we did a couple of work over here. So we started with young onset Parkinson's, and the subjects were from 18 years, and we gave a Saccad's task actually. So a reflexive overlap and reflexive gap, and similarly voluntary overlap and voluntary gap task. So this is how the tasks are presented in the whole thing. And the patient versus controls, and patient was, basically between the overlap condition, how the task was different. So the frontal lobe activation in patients in reflexive Saccad's task actually. And of course, this is trying to interpret the whole data. And we saw these areas basically during the Saccad's. This is in patients with respect to controls. That means it is a hyperactivation in patients. Mainly cerebellar areas were dominant, along with motor area, angular gyrus, cingulate gyrus, and middle frontal gyrus of the right hemisphere. And this is again the second, the overlap conditions that we tried to look at. And here the cortical activity was more in patients basically, and the voluntary Saccad's had more as compared to reflexive Saccad's. And the frontal and parietal were significantly more in patients in reflexive and voluntary as compared to controls. And another thing was the Saccad's is a reflection of the motor delay. The subject, basically everything boils down to the motor. That's how, not just this study, once we combined with a couple of more studies, we could say that it is due to the motor that the Saccad's are also impacted. So this is another study that we had done. So we had recorded spectrograms, and the spectrograms along with the functional data basically, and dynamic MRI. So we combined the three. We made the patients speak or vocalize during the functional MRI task, like the consonants, different types of consonants, whether it is velars, palatals, retroflex, dentals, bilabials, and nasals. So different types of sound, and all these were done. And we also recorded this dynamic MRI. So one slice cutting through, and then it was 40 milliseconds with the scanner that we had around that time. So every 40 milliseconds we were mapping, and we tried to see how during the vocalization, how is the tongue movement, or how is the acoustic cavity that is going to be there. And in many of the PD subjects, you would have seen that they are not able to speak initially, but once that Plosion happens. Once they are able to open that bilabials, they were able to speak without any problem, which was proven over here. The voice onset time, and it was directly correlated with dentals, bilabials, and nasals. And interestingly, I could answer the question of the neurologist whether I could differentiate between Parkinson's, MSA, and PSP using this combined approach. So we could differentiate between MSA and PSP, and also we followed up the study, and the 1 year follow-up was entirely different in MSA and PSP as compared to the Parkinson's disease. So this is the one. We also tried to estimate the acoustic cavity by measuring various parameters from these images. And basically, again, we tried to associate with motor disorder, and the M1 probably controls the velum palatini to produce the nasality, and the pitch was reduced in patients. As I said, only 3 category of the consonants were affected significantly, not the other 3. Now, we also tried, because one of the questions we had during that time was, is memory affected or impacted in Parkinson's? And we did a lot of studies. The study was very controlled, the paradigms were very controlled, and we could prove that there is no difference in the memory response. This was in off state. Off state is when they are off the drug for at least 12 hours. And on state is when the peak response of the drug after they take the medicine. So we had done the study in both the conditions, and so there was no change in the memory with respect to the healthy controls, and later it was proven by others also. They followed ours, and of course the corticospinal tracts and motor cortex, and we also tried to do volumetrics, and from volumetrics machine learning algorithms were used basically, and then we could show that substantia nigra plus thalamus could have good specificity and sensitivity, accuracy in differentiating PD from healthy controls. And we are now trying to do the same thing in differentiating the 3, now that our sample size is much, much higher. This another volumetric study which we did in mild cognitive impairment and cognitively normal Parkinson's. What are the areas that are affected? So in mild cognitive impairment, inferior parietal, angular gyrus, middle temporal gyrus, and middle occipital gyrus are much exhibiting atrophy as compared to cognitively normal subjects. So we also did domain-specific studies, and this is another thing that we attempted. And when the students or we sit with neurologists, we always encounter what are the possible things. And during one of the conversation, we found that some patients are having difficulty in differentiating the colors. So we actually did a detailed study where we used, of course, the resolution and all those things of the computing, the millions colors in the phone. So we tried to use some of them, and we differentiated. So the color perception task and task-based functional connectivity in PD basically exhibited alterations in left anterior temporal fusiform, right superior, and middle frontal gyrus. So basically these 4 networks were affected in PD, and so we could show that there is an impaired color perception in PD, and that could be because of the altered intratemporal connectivity. And again, we are not sure whether it is related to motor or not, because we didn't go into that direction, but then yes, definitely the color perception is impacted in them. And we also carried out olfaction. This was in the pre-COVID era. So we had a olfactometer imported from Europe, and but then it had a delay of 14 seconds. So we had used that, and we also used some indigenous method to study the effect of smell. We had used a sniffing kit, which is a very standard one from Germany. But some of the smells, like fish smell and all those things, were not acceptable to some of the patients. So we had to have our Indian smells. So we had taken 10 smells from India, and we normalized over that 4 smells, and we found that rose, orange, banana, and coffee were, they could distinguish that in the OPD. So this had the high probable values, and so we used it inside the fMRI, inside the MRI setup to study the functional effect. And you still see that the effect of smell in PD is much more limited as compared to the healthy. And of course, it is very significant in coffee. So coffee had the best activation in PD as compared to other smells. And so here again, between the functional connectivity study between right thalamus and left thalamus, there were significant alterations in PD as compared to healthy subjects. So we also did a ERP study, event related potential study, presenting the smell, and we show that, it has a possibility that it can distinguish PD, MSA, and PSP. So that is one possibility that it is giving. And interestingly, the subthalamic nuclei and cortical motor areas were affected with early motor symptoms. That is one more. So we also attempted neuromodulation, whether we can give TMS, and at that time, the study was initiated in 2014. So the TMS was attempted at 3 locations: motor area, and then the secondary, and then sensory motor cortex. And we found that, of course, we used functional MRI as one of the tools to evaluate, along with other clinical parameters. And so in motor area, we had the same subjects had underwent sham session and followed by real session after a delay. So we found that marked improvements in motor performance was seen after TMS in M1 and SMA, and sensory motor cortex did not have any such significance. Now, combining all the results that we had, somebody actually had used some of the conclusion that we had given, and they had supplementation of the amino acid, targeted amino acid, and they had shown improvement in the symptoms of Parkinson's. So that shows that we are going in the right direction. And it's a very big team. And without the collaboration of various faculty, like from neurology, neurosurgery, psychiatry, and ophthalmology, it would not have been possible. And yes, it would not have been possible without the students who are the main contributors to this study. Thank you very much.
Long live India and France friendship.